fortunate that we were able to move into the Morrison Life Science Center during summer of 2020. And we've been able to really expand our footprint and really expand our research, where we focus on engineering cells and materials for therapies and diagnostic and vaccination purposes. Well, welcome. This is my lab, or the Pannier Lab for Engineering Biomaterials and Gene Delivery. We're located on the 3rd floor of the north wing of the Morrison Life Science Center. And right now we're in what we call our main lab space, where we do a lot of research into designing materials to deliver genes or nucleic acids into cells, including stem cells, as well as for vaccine technology, similar technology that's used in the COVID-19 vaccines. And so this main lab is where we do a lot of our synthesis work, and as well as some of the prep work that we do to build up the DNA particles that we use and study in the lab. Not only are we really fortunate to be in the 3rd floor of Morrison with window views of campus, but we have access to a lot of high state-of-the-art facilities and technology here. And so we have fume hoods in every lab. We're able to have all of our instrumentation to make DNA and to prep cells. And so I just would like to highlight, we have a great amount of workbench space, and we are able to accommodate all of our students at individual benches, working on various technologies for stem cell applications and DNA delivery. So we're really fortunate that in addition to our main lab space, we also have a cold room space where we can store chemicals and perform experiments at 4 degrees Celsius. We have storage space and freezer space and autoclave, or where we sterilize instrument space. But we also have this space where we're able to perform cell culture, again attached to our main lab space. So this is where we can sterilely handle human cells and manipulate those cells and experiment with those cells to determine if our nanoparticles that we are developing are able to actually deliver genes into those cells. And so we actually have the capability of having 4 students performing cell culture in here at the same time. And we also have the capacity to have large incubator spaces and an imaging facility, a small imaging facility here, where we're actually looking at, these are stem cells derived from human fat that are actually growing and attached to the bottom of that flask. And so it's very nice that we can take the materials from our main lab and move them right into cell culture experiments in this adjacent space, also knowing that we have other spaces on the floor to do a lot of other applications and technologies.
As we continue on in the Iverson lab, you can see that we have a nice setup to do a lot of different chemical wet-based research. This is a pretty typical chemistry type lab. Over in the corner over here, we have 2 of my students, one graduate and one undergraduate student. They are working in the chemical hood. They are currently creating slides that have carbon nanotubes adhered to the surface. This sounds like it should be something that's pretty easy, but it actually took almost 2 years of research to figure out a way to get a nice even distribution of carbon nanotubes on the surface of the slides. And now the students are optimizing this process and using this process to create slides so that they can learn about what's going on inside the body. One of the great things about Morrison is we have this nice open lab space. And so we're now walking from the Iverson lab into the Kievit lab. Dr. Forrest Kievit is in charge of this section of the lab. He is also a professor in biological systems engineering, focusing on biomedical engineering. His lab uses nanoparticles that are sensitive in an MRI, and they are using it to learn about traumatic brain injury. There's so much we don't know right now about traumatic brain injury and how the damage that happens in a moment can progress over time. They are developing these nanoparticles to help us learn about it and treat traumatic brain injury as we go forward. The next lab in this group is Dr. Rebecca Walks. She is also assistant professor in biological systems engineering. with a focus on biomedical engineering. In her lab, they are looking at lower back pain. Lower back pain is something that afflicts a huge number of people every year. It doesn't have to be caused by instantaneous injury. It can be just life. A lot of people, especially in the agricultural field, end up with injury because they're constantly on a tractor. If you're on a truck, something that has little tiny vibrations constantly will cause degeneration in your spinal cord, in the discs that go between your bones. As that disc degenerates, nerves will sometimes grow into it, and that will cause extreme pain. We don't need those nerves there, and so Dr. Wax's lab is looking at ways to have the nerves fall back. So instead of growing nerves into a system, she's trying to stop nerves from growing, which is a little bit different than most people are doing in tissue engineering, but it's incredibly important, especially with the opioid crisis. People are in constant pain, and if Dr. Wax's lab can get this to work, they will no longer need to take pain pills. They will just not be in pain anymore.